So as Andres mentioned, I will tell you about what we did during these years to try to find a cure for Crigler-Najjar syndrome. You already heard about Crigler-Najjar syndrome. Crigler-Najjar syndrome is a genetic defect of liver metabolism and is due to, is related to bilirubin. Bilirubin is the main degradation product of heme metabolism. Through a series of reactions, heme is converted to biliverdin, that is water soluble and is not toxic. However, Mother Nature decided to produce another compound that is unconjugated bilirubin that is toxic and insoluble. And usually this is converted to a soluble compound that is conjugated bilirubin through a reaction that is driven by UDP glucuronide transferase 1A1 that attaches two glucuronic acids to the unconjugated bilirubin and renders it soluble. However, when this enzyme here that is mainly expressed in the hepatocytes of the liver, when mutations affected the gene, the UGT1A1 gene, this enzyme is not working or is working less, and so you have an increase of unconjugated bilirubin that accumulates in the bloodstream and deposits in lipid-rich tissues, such as the brain. Crigler-Najjar syndrome is an ultra-rare disease affecting 0.6 to 1 per million live births and is an autosomal recessive mutation of the UGT1A1 gene. The gene is composed by different first exons that are located here in the 5' and 4 common exons that are spliced together with the first exon to generate different mRNAs. Every first exon is specific for a specific substrate. The 1A1 is the one responsible for bilirubin conjugation. So mutations in the UGT1, in the 1A1 or in the common exons affect UGT activity. Depending on the mutation, you are going to have different activities of the protein from 100% no mutations to 50 or less to up to 0% of activity, and this reflects also bilirubin levels. So when you have 100% activity, you will have bilirubin levels that are below 0.5 milligrams deciliter, and in the range, if you have no activity, bilirubin goes sky high and is about 20 to 50 milligrams deciliter. This is life-threatening and needs to be treated, okay? So there are two different types of, this is called jaundice. So when you are yellow because of bilirubin, this is called jaundice. And this particular disease appears as early as the first week of life and is characterized by a yellow discoloration of the skin, of the eyes, and babies, if not promptly treated, develop brain damage, okay? So, and they can die because of kernicterus, which is the manifestation of bilirubin toxicity. So, available treatments are, first of all, phototherapy. So babies undergo 12 hours per day phototherapy. They just lie under this blue light. Blue light is able to convert bilirubin to a water-soluble photoisomer that is easily excreted by the body. And once the child grows, parents, they have these special devices that are put on the bed, and so they sleep 12 to 40, or they have to stay under the phototherapy for 12 to 40 hours per day. The problem is that during growth, the skin gets thicker and phototherapy is not anymore effective. Of course, there are, during the years, technicians have developed also some portable devices So these kids or adolescents can go also on holiday because they have these blue light LEDs portable, and they can handle not an easy life, but is almost feasible. However, as I was mentioning to you, phototherapy is not effective for long periods of time, and also concurrent diseases, also a fever can increase bilirubin levels, so these patients have to really stick on phototherapy. And once phototherapy is no longer effective, they have to go undergo liver transplantation, which is, of course, is not easy, is not devoid of risks, and moreover, they have to undergo liver immunosuppression all the lifelong. So our goal was to find new therapeutic approaches to cure the disease. And I would like to tell you, of course, about experiments, but also about meet the right person at the right time. So the story of my research. So everything started for me in Crigler-Najjar Syndrome when I joined the group of Professor Tiribelli at the Liver Research Center here in Trieste. I was doing my PhD and I was studying bilirubin neurotoxicity, how to tackle the disease. And I was working with gun rats, which are a rat model of the disease. Unfortunately, this model is valuable, but doesn't die. So it was difficult to assess how bilirubin was affecting and the outcomes also of treatments. So one day, Professor Tiribelli called me in his office and he told me, well, Giulia, I have a wonderful idea for you. You're going to do a mouse, a knockout mouse. And I said, well, this is a very nice idea, but I don't know how to do it. So I started studying, and molecular biology was not an easy task for me, especially because in the lab we didn't have the possibility to do molecular cloning. So it was tough, and one day I was at lunch with this crazy and excellent researcher, that is Ennio Tasciotti, and I asked him, well, Ennio, you have to do a knockout mouse, but I don't know how to do it, so I'm messed up. And he told me, well, I know the right person for you. So he made an appointment with this wonderful mentor here, that is Andres Muro. And I met him, we discussed, and he said, okay, yes, come to my lab and we're going to do the mouse. So what we did was a gene targeting approach, a classical one, generating a one-base deletion in the exon 4 of UGT1A1. And we did it, the same mutation that was present in the rat. But remember, the rat doesn't die. And because we were afraid that the single one-base deletion was not sufficient to generate the phenotype, or a strong phenotype, we also flanked the exon 4 with LOXP sites to remove the entire exon 4 in the case in which the single base deletion that causes a frame shift was not sufficient. Of course, because of gene targeting needs, we also included a neomycin cassette and the TK that was useful to exclude non-homologous recombination events. So this molecular cloning took us pretty a long time, also because I was not so good in molecular biology, and then because we realized that UGT1A1 has also a pseudogene. So this pseudogene was making our analysis more complicated, so we decided to elongate a bit the screenings just to be really, really sure that everything was right. Then when once obtained the ES cells, the recombinant cells, we generated the chimeras. The first round of chimeras that we generated were not transmitting, so we were so anxious because we were working so much, and indeed we didn't know if it was effective or not. We did a second round of chimeras, and we generated 10 chimeras, and all of them were transmitting the transgene, the mutated gene, and we were pretty happy about that. And then we crossed these animals, we removed the neomycin cassette, and finally we got the mutant animals. And the mutant animals were yellow. So, meaning that all this work was fruitful. So we were very excited, and we started the analysis. So what we saw is that, of course, jaundice was due to the absence of the UGP gene and no activity of the enzyme. As you can see here, is conjugated bilirubin in a wild type and in an hetero, but no conjugated bilirubin in a mutant. We did bilirubin levels, and we observed that bilirubin levels were already sky high at 2 days after birth, about 12 milligrams deciliter, and they were increasing over time. Unfortunately, or fortunately, we would say, we will see later on why, these animals were dying as early as 1 week after birth. So after all this work, the animals were dying. So we were pretty desperate because we could not study so many things. But at that time, yeah, of course, they were dying, and they were dying because of bilirubin accumulation in the brain. Here is the brain of a mutant animal, and here is a wild type. As you can see, it's orange, full of bilirubin, and affects also brain morphology. You see, this is the cerebellum of a mutant animal. That here you can see the arborization of the cerebellum is completely different from a wild type. We saw that cells were dying by tunnel analysis, and further we also characterized the molecular events associated with this cell death. And we observed that cerebellar cells, in particular Purkinje cell layer, there are these green cells here, were really affected in their number and in their dendritic arborization. So we were satisfied from one side because the animal recapitulated the disease, the human disease, but animals were dying very fast. At that time, AAV gene therapy was rising and was spreading, and as you already heard in this course, AAV gene therapy is a powerful tool to test, to try to cure monogenic diseases, and this relies on the capacity of these small parvovirus to carry gene expression cassettes of about not that big, but enough to insert a cDNA under a transcriptional control of a tissue-specific gene. And you can also pack them in the DNA into different capsids to trigger tissue-specific tropism. Okay, for example, AAV8 and AAV9 are very good for liver targeting. AAV9 is very good for heart, and skeletal muscle has different serotypes. So, and the very promising results of gene therapy were coming from different phase 1 and phase 2 trials for different diseases that at that time were nicely recapitulated by Federico Mingotti in this review, and also by Serena Zacchigna, in which you can see that Among the AAV clinical trials present up to 2014, the majority was about monogenic disorders. Okay, so Andres said, so why don't we try to rescue the animals with gene therapy, with an AAV gene therapy? So we said, okay, let's try. And this was also possible because at the Institute we have an AAV facility that is headed by Lorena Zentilin with the approval of Professor Mauro Giacca, and these very helpful technicians that are Marina and Michela that produced the first vector. So we cloned the human UGT1A1 gene under the transcriptional control of CMV promoters, so to express the gene all over the body of the mouse, and we used AAV serotype 9, and we injected animals at postnatal day 2. We monitored survival, and we observed that the animals treated with gene therapy were all rescued. So, meaning that this gene therapy was successful, and these animals lasted for 5 months at the time in which we decided, okay, let's sacrifice the mice and let's see what's going on. And what we observed is that bilirubin levels were really low, indicating that it was very effective. And next, of course, we also analyzed neurological damage to assess if this gene therapy was able to rescue not only the animals but also the neurological damage, and yes, it was. It was able to completely restore the cerebellar architecture that we used as our marker for efficacy, and also Purkinje cells developed normally, the same as a wild-type animal. So we were pretty excited about that, but what surprised a lot us was that expression was not localized in liver, but was extra-hepatic, in the skeletal muscle. Okay. Here is a GFP staining to show you that the muscle, but not the liver, was expressing the transgene. And here you have the Western blot at 15 days post administration of the virus and 150 days after, and only the skeletal muscle was expressing the UGT1A1 gene. Nice, but UGT is not expressed by a skeletal muscle. So what we wanted to do was to achieve a liver-specific expression. To do so, we used a serotype 8 to achieve nice transduction of the liver, and we used alpha-1 antitrypsin as a driver for the expression. Alpha-1 antitrypsin is a liver-specific promoter. We injected the animals at P4, so a bit later, and we monitored survival. Again, now we show that nicely the liver is transduced and the transgene is expressed, so very good. While skeletal muscle, no, it's not in these slides. Another very nice finding was that treatment was effective 48 hours after the delivery of the virus, so very fast. As you can see, here is a mouse, a yellow mouse, and this is 48 hours later, pink. And this is the quantification of bilirubin levels. So it was great. We were pretty happy. And this animal lasted for 17 months. This is the bilirubin levels over time. And as you can see, over time, we observe an increase in bilirubin levels. This green bar here are the animals treated with the gene therapy. But still 50% less bilirubin compared to an untreated control. So, nice. Why we observe a loss of efficacy over time? This is normal because we are treating the animals with an AAV vector that is an episomal vector, so it doesn't integrate. And once the animal is growing, the liver is duplicating, and you observe a loss of genomes. This is due to several factors: dilution, loss of DNA. So Up to 2 months, the levels of episomes is stable and lasts for the entire life of the animal. We also demonstrated that it was not about integration because we could observe high molecular weight episomes and double-stranded circles in the liver of the mice. Of course, the drop in viral genomes was mirrored by a drop in UGT activity, retaining still about 8% of activity 2 months after the injection, and this was confirmed also by Western blot. Nice. Another curious thing was that when we ran together liver samples, so animals treated with a liver-specific gene therapy, together with the animals treated with those animals that were expressing in the skeletal muscle, we observed that the skeletal muscle was expressing much more UGT1A1 than the liver, about 2.5 to 5% in the liver compared to 10 to 30% in the skeletal muscle. However, bilirubin levels were much lower in the animals treated with gene therapy. Gene therapy liver specific, indicating that extra-hepatic expression is okay, but is not as good as the liver specific. And we evaluated 2 main transporters in this context that are MRP2 and MRP3 that are present in the liver but not in the skeletal muscle. These transporters are able to remove bilirubin from the cells, and they send it to the bloodstream or to the bile. And so once bilirubin is conjugated, it goes out and it's excreted. So probably that's the reason why this is happening. So for the first part, we can conclude, of course, that the mouse that we generated actually recapitulates very well the human syndrome. So the animals die, they have neurological damage. We can also say that extra-hepatic expression is okay, although it's not efficient as the liver-specific. However, liver-specific expression faces limitations because hepatocytes are duplicating and you lose vectors. So we, together with Luca, a former PhD in our lab, we tried a third approach, double injection. So a first injection with an AAV vector serotype 8 very early in the neonatal period, and then a second injection with AAV9 in the adult period. We changed the serotype because during the first administration you can generate neutralizing antibodies against against the capsid. So the second administration might be hampered by these neutralizing antibodies. We tested this approach by 2 reporter genes, GFP and mCherry, and as you can see, the serotype switching is effective. As you can see, when we administered with the second injection AAV9 mCherry, the transduction is very similar to only a single administration of AAV9, indicating that there's no cross-reactivity between the 2 serotypes, and this is important. So the idea was very robust, and we tried with UGT. And of course, what we observed is that the double injection, the blue line, was more effective than a single neonatal injection. Nice. Wild-type levels are here, green, comparable to our double injection. Again, we observed in neonatal single gene transfer a rise in bilirubin levels over time. Interestingly, when we went to the molecular analysis, we observed that the double injection doesn't reach actually the same UGT levels compared to the adult single injection, but this probably is due also to injection procedures. So, never mind. What was really scary was the fact that when we look at the liver of the double injected animals, we saw liver anomalies, indicating that these doses that we were giving to the animals were not good for safety reasons, okay? But single neonatal or adult injections were safe. So here it rises, of course, the need to lower the dose and so have a much potent vector. So a potent vector gives you the possibility to reduce the dose, okay? So we will see later how we did this. Another important thing that Luca observed is that when you transduce, and here again we go from males to females, when we transduce males, the transduction efficiency is very nice, while when we transduce females, transduction is not so good. And this is known and is related to hormones. So if you treat males with female hormones, you get the same result, and vice versa. If you get females becoming more male, they transduce better. So it's a matter of hormones, but not only. We will see why later on. Another important issue was that when we injected male animals, treatment was effective, lifelong, 15 months after administration, while in females we observed a drop in bilirubin levels as early as the treatment was performed, and then we observed an increase in bilirubin levels, indicating that we were losing efficacy. And this was due also by a rise of immunoresponse specific in females but not in males. And this was detected by different markers of inflammation and also by interferon gamma evaluation in dendritic cells. So another important issue that arose was the targeting non-hepatocytes. Even though we were using a serotype that is specific for liver, and also a promoter that is specific for hepatocytes, you cannot rule out that you are targeting also other cells. And the researchers during the years show that if you target antigen presenting cells, this is a problem because they will present they will trigger an immunoresponse against the protein. So Luca inserted a microRNA decoy in the AAV vector that is able to bind to microRNA 142 and to abolish expression in antigen presenting cells, because this microRNA is specific for antigen presenting cells. He tested this ability in vitro and it was okay. So then we produced the virus and we tested it and see what happened. What happened? In males, nothing. From the point of view of bilirubin levels, the efficacy was the same. In females, again, it was not a matter of the targeting antigen-presenting cells. Females still lost their efficacy. But what we observed when we went to the molecular analysis is that indeed, when you target antigen-presenting cells, UGT181 expression is decreased compared to a microRNA decoy that is not binding, so inverted. Here you can see the difference. So meaning that a certain degree of effect is present and it's important, although this is not related to the differences that we see in females. So after these studies, we concluded that serotype switching is okay and it evades the AAV cross-reactivity, and it's possible, but you have to have a very potent vector to decrease the dose and avoid liver abnormalities. Adult injection is efficient in males, but not in females. And that even though the targeting of antigen presenting cells didn't improve the efficacy of females, reduced the expression of males, indicating there is a certain extent of effect on this. All these studies were done with a lot of efforts, and actually we were new in the field, and it was pretty difficult to be accepted. But indeed, one day, a man, a very excellent scientist, that is Fulvio Maviglio, came here to give a presentation. And at that time, he was director of Geneton in France. And he was presenting their fantastic results for Duchenne. And Geneton is an institute in which they work for gene therapy to find a cure for different diseases. At the end of the presentation, he listed different diseases they were actually trying to tackle, and among them there was Crigler-Najjar. So after the presentation, me and Andres, we went to him and said, listen, but we are working on gene therapy and on Crigler-Najjar, so maybe we can collaborate. And he said, well, let's collaborate. And he put in contact us with this excellent researcher that is Federico Mingozzi, that at the time was, and he is still part of Geneton, that was organizing a clinical trial for Crigler-Najjar syndrome. So we joined this wonderful group that is spread across Europe, and it gathers together different centers from UK to Naples, all around Europe. And we started this adventure to find a cure for, to find a way to achieve a clinical trial for Crigler-Najjar. And the first thing that we did together with them was to optimize the vector. So this was done by optimizing the cDNA, the human cDNA, by different algorithms. We came out with different vectors. Here I show you just two of them that were modified to find the one that was expressing better the gene. We also modified the vector per se, removing cryptic splice sites that were reducing the expression levels, and indeed we observed that removing cryptic splice sites was very effective. We tested these vectors in two different models. Our model, the neonatal UGT, the little one, and again the gun rat, because having different models, it's always useful when you want to show that something is effective. And we observe, of course, that this vector was very effective in rats, in adult rats, and lowered also disease bilirubin, conjugated bilirubin in bile. So increasing the dose of the vector, we observed an increase in conjugated bilirubin in bile. We also showed that the vector was effective in neonatal mice, and this was good. We tested many other things. So one of these, for easiness, I just list them because otherwise we can stay here forever. Not forever, I'm joking. Since you have two options when you have AAV vectors, you can have a single strand, let's say vector, or a self-complementary, in which basically two cassettes are in the same vector, and you can achieve higher expression levels with one single particle. So this is very good. However, when we produced these single-stranded, was these are empty particles and these are full particles. You can see this band in the cesium gradient. This was a very clear and nice band, very abundant, while when we were performing single self-complementary, this band was faint and was not produced very good, so we decided to go ahead with single strand. But this is just our case. Sometimes self-complementary is better. We also optimized the production method. Of course, we were working with mice and rats, and this usually you produce the virus using adherent cells, but when you have to treat a patient, you have to have bioreactors, so supernatant cells. And we observed that when we produce as adherent or supernatant, the effect on total bilirubin levels is the same, the vector is okay, supporting the production method in bioreactors. We also did GLP toxicity, biodistribution, and shedding to assess that the vector was not toxic to the animals. Toxic in two ways, toxic for the per se, for the vector, but also for the production purity, because if we want to go to a patient, we need to be really sure. Another important thing that we were investigating was that, of course, we were studying the human version in a rodent model. So how we can compare it, and how we can exclude that what we observe in the mouse is due to a human version of the protein and not for the protein per se. So we did some immunogenicity studies, and what we observed is that if we treat the mouse with his own mouse cDNA of UGT, we have also, this is very effective, and we have also the rescue of females. So, meaning that to a certain extent, the loss of efficacy that we were observing in females is due to immunoresponse against the human protein. And none of the animals treated with the mouse, the specific version, so the mouse in mouse, was generating antibodies against the protein, his own protein, while the majority of the animals were generating an immunoresponse against the human protein. This was also verified in rats and was the same. So this indicates us that once we go to the patient using the human version, we're going to have a good chance to succeed without immunoresponses. Next, what we wanted to investigate is, of course, if we treat adults, now we know that basically everything is going well. But what happens, if you remember, when you treat neonates, you have loss of efficacy. So which is the best time point to treat, to have long-lasting expression? So we decided to take different ages, P4, P11, and P18, and we administered the vector, now codon optimized, super vector, purified, wonderful. And what we observe is that, of course, the more you wait for the liver to develop, the better it is for the correction of bilirubin levels. As you can see, P18 is much better than the other time points. Here is an immunofluorescence of UGT in green, and glutamine synthetase in red to show that once you administer it, the green is brighter and more frequent in juvenile animals compared to neonates. Next, we also investigated if this was a short-term experiment in which we wanted only to see which was the best time point, and then, okay, selecting P18 as the best age, we monitor bilirubin levels over time. We did a dose finding to find the dose that was able to correct bilirubin over time, and this was achieved with 5 10 to the 12 viral genomes per kilo. Nice. Correlated well with viral genomes and with UGT expression. We were satisfied. After that period, we sacrificed the animals and we checked for liver abnormalities, and we could not detect any liver abnormality at the low and the high dose, indicating that the vector is doing its job very well and is not triggering any effect in liver parenchyma. Next, of course, the neonatal experiments were done using IP injection, but we not think about an IP injection in a human. So we compare different routes, so IP versus retroorbital, that is a kind of IV injection. And of course, we observed that retroorbital is much more effective than IP injection, supporting and at lower doses, supporting this pediatric investigation plan. So overall, we can say that the optimization of the vector, if we compare it to the one that we were using at the beginning, is about 100-fold more effective than before. We developed a scalable process to support the use of bioreactors for the production of the vector. We did safety studies with showing no adverse effects, both in rats and mouse and also in rabbits. Immunogenicity studies revealed that there is a species-specific immunoresponse against the protein that would be avoided when we are going to be human in human. And of course, juvenile studies are ongoing to model the optimal parameters for a pediatric gene transfer. So, about the clinical trial now. So the clinical trial, it was a long work also in this context that belonged more to the clinicians, in which they identified the patients, their mutations, the identification of who was eligible or not to join the clinical trial, the screening of pre-existing AAV antibodies. Of course, if you already have pre-existing antibodies, you cannot be enrolled at the very beginning of the clinical trial because the gene therapy will fail, so it's not our purpose. And of course, who is enrolled undergoes an observational baseline of TB levels to understand how bilirubin levels go before the treatment, to understand if everything is going well. The first patient treated has been treated in December last year, 2018, is Melissa. She has 22 years old. And a second patient has been treated in the Netherlands in February, more or less. And now we are waiting for the third patient in Italy, so very soon. Yet I cannot tell you anything about the results of Melissa, but we are very positive. And this is the people who made it possible, the CureQN Consortium. It's a lot of people and gathers together clinicians, researchers, patients associations, and from different parts of France, Netherlands, Italy, and a lot of efforts. So I would like to conclude thanking all the people that took part on this very exciting work. And I would like to tell you one last thing, that you have to struggle a lot and to work a lot, but you have also to be surrounded by very good and the right persons to achieve your objectives. So good luck to all of you, and thank you for your attention.